Hello, this is Mickey Urdea from Haltaris Associates, and welcome once again to another episode of Interesting Stories in the History of Diagnostics. Today I'm going to talk about the plague, the Great Mortality, the pestilence, and sometimes referred to as the Black Death, although that term Black Death goes all the way back to Homer, who in the Odyssey actually referred to the monstrous Scylla as she with a mouth full of black death. But that didn't have anything to do with the plague. And in fact, what the first plague actually was has been a controversy for quite some time. And I'm going to address that a little bit here later today. So we now know that the plague is actually a often fatal disease caused by the bacterium Yersinia pestis, pestis being related back to pestilence. But the genus name, Yersinia, comes from an individual named Alexander Yersin, who was in fact a student of Louis Pasteur and was involved in a study of the outbreak of plague in Hong Kong in 1894. It's been around a long time, and it's still with us, as a matter of fact, as I'll come back to that as well. There have been a number of open issues concerning the plague for centuries, one of which is what caused it. We now know that's Yersinia pestis, but that is actually related to the question of what was the first plague, and that is, when was that particular organism associated with a disease outbreak? And as you'll see, there have been many such outbreaks over the centuries. But we've got a new tool that has been very important in trying to settle many of these questions, and that is DNA analysis, DNA sequencing, clade analysis, and all the information that generates for us. And you'll see how that's being used to answer some of these questions in this episode. A lot of the things I'm going to talk about have been very recent. In fact, 2021, 2022, the earliest one going back to 2013, and that's because we now have finally developed the appropriate DNA analysis tools that permit these answers to be made. These new technologies are making it possible for us to study ancient DNA from humans, from the organisms themselves, from even the rodents. And you'll see that this is all being rolled up now to learn much more about the origins and the evolution of the disease. I'm going to give a little bit of background on the plague. I'm not going to go into the standard issue of the symptoms, the buboes, all of the other things about the disease itself. I want to spend more time on the actual history here and talk about how the DNA analysis was helpful in understanding it better today. So the plague that is referred to as the Black Death, which wasn't actually called that until 1755, actually ravaged northern Africa and most of Europe between 1346 and 1353. During that period of time, a large number of people died. Between 75 million and 200 million people are likely to have perished in that outbreak. Reportedly, the first appearance of the disease in Europe occurred with the siege of the Crimean port Kaffa, and the city was attacked and sieged by the Golden Horde Mongolian army, headed by Johnny Beg. And during that particular assault was one of the early occasions of so-called biological warfare, where they actually catapulted bodies that were known to be ravaged with the plague over the walls of the city to spread it within the city. Unbelievable. After the siege of Kaffa, the Genoese galleys once again were sailing. They took off and went to Sicily. They went to Genoa. They went to Venice. They went to Marseille. And from those ports, it spread north into Europe very rapidly. Europe was poised for this spread. It was an incredibly filthy place at the turn of the 14th century. The streets were filthy. The people were filthy. For instance, St. Benedict actually said, "To those who are well, bathing shall seldom be permitted." And in fact, St. Agnes, who was a big proponent of what St. Benedict suggested, actually died without ever having bathed. The streets were disgusting. As a matter of fact, Italian cities and also French cities started actually naming many of their streets after names for human waste. For instance, in Paris, there were streets named Rue Merdeux, Rue Merdelet, Rue des Mordans, amongst many others. Many of them are very interesting. In fact, they actually had another avenue called Rue de Pipi. So it was filthy. Everyone knew it, and even there were jokes about it. But this just set the stage for the rats that are coming in and the fleas associated with them. It was perfect for incubating this incredible disease that spread so rapidly to so many people. As a matter of fact, it appears as though 40% or more of the population died, and it was quite variable from one place to another, impacting 75 to 200 million people, although the actual numbers will never be known because, as you might imagine, with so many people dying, records really weren't kept in a way they had been in the past. As a matter of fact, the population of Europe in 1300 really never came back until approximately the 1500s. So now I want to talk a little bit about how DNA analysis has been used to learn a lot more about the plague. So, for instance, I said earlier, what was the first plague? What was the first plague that was associated with Yersinia pestis infection? And we now know that that was what is referred to as the Justinian plague. This occurred in initially Byzantium, the eastern part of the old Roman Empire, under the Emperor Justinian, and it spread from that area to most of the globe. It was a devastating, broadly impactful plague, which had been known for quite a long time, but nobody really was certain about what caused it until, in fact, 2013, when samples from people that had perished in that particular plague were analyzed, and there was clear evidence of Yersinia pestis infection. And also, DNA analysis has been used to verify the fact that the second plague, now the Justinian plague is called the first plague, and now the Black Death is called the second plague. The second plague was also the result of Yersinia pestis infection. We've learned a lot more about that I'm going to come to in a little bit as well, which tells us a little bit more about entry and how old that plague is. That second plague didn't go away for a very long time. As a matter of fact, it really didn't go away until around 1850, believe it or not. In the Islamic world alone, there was one outbreak of plague in at least one location every year between 1500 and 1850. Amazing. In Istanbul alone, there were 68 outbreaks between 1700 and 1800. It was everywhere. I mean, it just didn't really go away. There was a gigantic bolus of disease very early on, and then continue outbreaks all over the planet for the next several hundred years. There was a third plague, and that one initiated in China in 1855 to 1859. And that one spread globally as well. As a matter of fact, more than 10 million people died in India alone. So this went on, believe it or not, till nearly 1925. In fact, there were even outbreaks here in San Francisco. There was an outbreak between 1904 and 1906, and another one in 1907 to 1908, kind of before and after the Great Earthquake. So it really hasn't gone away since the very early days of the first noted Yersinia pestis plagues. It is less so now, but we still have it all over the world. In fact, there was an outbreak in 2017 in Madagascar. 170 people died. The mortality rate has gone down dramatically for a number of reasons. We have insecticides to deal with the fleas. We've got antibiotics that work quite well. We also have a vaccine for plague. However, it's a little scary that there was an outbreak in 2009 that was associated with a known now resistant form of Yersinia pestis against most of the known antibiotics that were used. So that's a bit of a scare. There are a few very recent, very interesting studies that put a lot more light onto the origins of the Black Death in Europe. Remember that I talked a bit about the Mongol army and attack of Kaffa. That suggested that the plague was coming from somewhere east, somewhere in Asia, Central Asia. There were even reports that it might have been from China, but nobody really knew at that point. That started to clear up in 2020 with the report of a study conducted at the University of Warwick, where they looked at over 1,300 samples of Yersinia pestis that have been collected from all over the world at many different times, and they were able to piece together a family tree of Yersinia pestis over a 5,500-year period, including strains that are before any known plague. So this was really quite a remarkable feat. And one of the things that was very clear was that there was a rapid diversity of the Yersinia into 4 new strains very early in the 14th century. Very interesting situation, where that's when we started seeing the second plague take off. One of those 4 divergent strains that came about was actually responsible for the Black Death in Europe. Now, researchers recognized that that was not the mother strain that led to the explosion, because there were 2 mutations in that particular genome that were not found in the mother strain or even close to it. There was reason to believe that that mother strain came from Central Europe because it was found that the rats, even today, actually harbor Yersinia pestis that's much closer related to that than the European strain. Then in 2022, the historian Philip Slavin from Stirling University in Scotland had been looking at a series of burials in a graveyard in northern Kyrgyzstan that were associated with a trading community, and these dated back to 1338. It appears at that time there was a pestilence which occurred in that area and took the lives of quite a few people, but no one actually knew what it was that they were infected with. There were a lot of different pestilences around at that point, so it could have been Yersinia pestis associated, or it could have been a number of other things. So Slavin had the idea, well, we ought to take a look at the DNA. Fortunately, the physical anthropologist Barbara Bamonte of the University of Ferrara, and then Maria Spyrou, who is a paleogeneticist from the University of Tübingen, heard Slavin give a talk on the work he did on the burials. And they got together afterward and started a collaboration. Working along with a Russian team, they actually got permission and were able to use the teeth of 7 of the victims of the 1338 outbreak and analyze DNA within them. And they looked at 7 particular teeth from 7 individuals, and 3 of them actually had Yersinia pestis, which is kind of amazing. So clearly, this was an early outbreak of plague. That's what the pestilence was. But most interestingly, the particular strain that they had was right at the base of that explosion of 4 different strains that occurred in the 14th century. So this was early. It must have come from Central Asia. They pinned it. And this is now nearly a decade before the outbreak in 1347 in Kaffa. The mystery of the spread of the disease still remains a bit controversial. And I think there are 2 different parts of this. One is in Asia before it spread to Europe, and then after. Now, within Asia, people initially talked most about rats, but also now marmosets and voles are brought into the picture. And then my favorite is the gerbil, which many people think is probably the one that was moving it around within Asia before it actually got into Europe. It's probably a different picture after transferring to Europe. For instance, actually, DNA of rat bones has been able to show fluctuations in rat populations in Europe and Asia over long periods of time. And at the time of the plague, there were the same species of rats in Europe and in Asia, and those species spread within the European population during the plague. So it seems much more likely that the fleas on the rats in Europe were at least one of the causes of spread, but the other is probably person-to-person transmission. Many researchers now believe that the aerosols from pneumonic plague were being spread by one person to another, not just infections based on the bites of fleas. By the way, one little tidbit on how fleas actually infect people. It ends up that Yersinia pestis actually blocks the gastric system of the flea, and they don't process their food properly, which of course is blood. So they try to get by this by actually regurgitating their food, which also contains the Yersinia pestis, and that then generates its transfer from the flea into the victim. So very interesting little piece of biology there. So now at least we know that the plague goes back to at least 1338 and in Asia, but who knows how much further it goes back? It might go back centuries. Who knows? We'll find out. Another series of studies also reported in 2022 were based on the notion of trying to understand why was it that some people died from the plague and others didn't? Is there potentially some genetic difference in one part of the population versus another that permitted some people to survive and others not? So there were 2 studies reported, one in London and another one in Denmark, both of which were looking at people that had died before the plague, people that died right at the height of the plague, particularly in 1347 to 1348, and then afterward, and trying to see if there were differences amongst them. It's interesting that they found a significant difference in a particular gene, which is referred to as ERAP2. And this gene is related to the ability of the human innate immune response to hold off viral infections. So they were curious about this observation, and a series of studies were conducted on cultured human cells that were grown in the presence of Yersinia pestis. And interestingly enough, like with viruses, Yersinia pestis growth was also suppressed by the presence of the UAP2 mutations. In the London study, they found that approximately 40% of the people before the plague harbored these 2 genetic mutations, about 35% of the people died during the plague had it, and it rose over 50% after the height of the deaths in the second plague. More interestingly, but in a smaller set of samples, in a Danish study, about 45% of the population had these mutations before the height of the deaths during the plague, but about 70% afterward. So much bigger difference. Though this is not the smoking gun, it's not proof, but it's very interesting. And that group of investigators is now looking at additional variants, other genes as well, to see if they can't understand more fully whether or not there were people that were just inherently more susceptible to die from plague than others. So it's just amazing how much has been learned in just the last decade about the origins of this disease and its spread and where it initially occurred. All this being done by our ability to isolate these ancient samples of DNA, which of course was the basis of one of the Nobel Prizes last year with Fonte Pabo, ability to actually analyze the data, which is not that trivial, to put them into clades and into an overall family tree where we can understand what's transpiring over time. We're going to see a lot more, and I just can't wait to see what else comes out as a result of these kind of studies. I've talked a bit before about the application of DNA analysis to look at other diseases. For instance, I talked about this with regard to the history of syphilis. We know more about TB as a result of this. We know a lot about a lot of different disease states by actually looking at the presence of the particular organism and its actual DNA fingerprint to understand where it is that it came from, how it's changed over time, and how it relates to the organisms that are present in the population today. And of course, we're seeing this right now in the case of SARS-CoV-2 infections and COVID-19. That's been an extraordinarily important bit of information for us to understand on an ongoing basis around the globe to see the transmission of the various forms, alpha, beta, Omicron. So these technologies are applicable today, and they're applicable in looking at samples from ancient outbreaks and learn a lot more about those diseases. And who knows what implications they're going to have in terms of our understanding of how to deal with those diseases as they show up again and again over history. I hope you enjoyed this, and thank you for tuning in once again in another episode of Interesting Stories in the History of Diagnostics. This is Mickey Urdea for Halteris Associates, and thank you very much, and bye.